Hello, Marissa. Welcome. Thank you so much for agreeing to give us a user opinion. To all the viewers, this is Marissa Baker from Genesis, and she will tell us more about how FIDA is being used by Genesis in their work with de novo proteins. So, Marissa, would you tell us a little bit more about yourself, your role, and what does Genesis do?
So thank you so much for having me. My background is more biochemistry and biophysics. My group is responsible for all the biochemical and biophysical screening going on at Genesis here. So all of the cell-free assays are in my group.
Hmm? And Genesis has this special AI operating system for drug discovery called GEMS. Would you tell us more about it?
Yeah, so GEMS is basically our own one of our internal tools where we're doing virtual screening. So kind of our pipeline is to do virtual screening on a target. It kind of generates a lot of options, and then we rank those options using our internal AI tools. And then the chemists will go through and they'll, again, rank order with an actual human being. And then they'll choose compounds to either synthesize or purchase, and then those go into the real assays, whether it's our biophysical assays or cellular assays. And that kind of generates the real data that will then go back into the model.
So there's a feedback loop
Yeah.
involved. Okay, that sounds very efficient to me. And how does FIDA fit into the workflow?
Yeah, so FIDA is one of the biophysical tools that we just brought on very recently. And so we've been using it as more of a screening platform, kind of like an in-solution method that we weren't really able to use before to generate KDs. And so we're either using it to um, confirm a different data that we got from a different biophysical method, such as, SPR or thermal shift assays, and just make sure that the data is correlating, or our cellular assays will give us some sort of functional readout, and then we'll use FIDA to get some KDs that correlate as well.
Mhm. So you find that the in-solution character of the technology is a real advantage in your work?
Yeah, that's what brought me onto it originally. Some of the current in-solution methods, at least that I'm using, either don't generate as consistent data as we would want,
Mhm.
or they use a lot of material, or it takes a very specialized technique that we were having trouble either bringing in-house or using kind of an external source. And so the FIDA was exciting to have more of a quicker method that gave pretty reliable data and was in solution. Yeah.
Yeah, okay, perfect. So yeah, I see where it fits and why. That's interesting. Okay, so it did help you to overcome a couple of limitations that you were facing. Um, is that right?
Yeah, so truthfully, SPR is kind of our workhorse here, and it always has been. But there were a couple proteins that we could never see any ligands binding by SPR, even though we knew they were binding from other methods, even structure. And so we were kind of perplexed as to why they weren't working. We just assumed maybe the protein reagent just wasn't going to work, maybe for a biophysical assay. And then once we ran it on the FIDA, we actually quickly saw binding that correlated with our biochemical data and cellular data. And so we realized
Mhm.
that in solution is actually more important than we realized.
Okay, so you work with difficult targets then and new modalities as well.
Yeah, as a company, I think now since we have this AI to leverage, we are choosing difficult to drug targets, at least in the eyes of everybody else. So some of our protein reagents are generally hard to make. There's not a lot of precedent for them. Some of them have to be in a specific complex. So one of the advantages of FIDA is also that you can tell what the molecular weight of the proteins that you're testing at the concentration that you're testing, which has also been a huge advantage for us here.
Mhm. And what about the sample consumption and the preparation it requires?
Yeah, actually, so we are doing Sci5 labeling of all of our proteins since we are a small molecule company. Um, if you just read at the UV range with the proteins, there's often interference with the compounds. So when we label all of our proteins with Sci5, that does use up a little bit of protein to make the stock. And so it's really nice that you only have to use a very small amount of protein when you're actually running the assay, because once you label the protein, you have a stock that will last quite a long time. So it's a nice way to not have to use up all of your protein, especially when it was a hard to make one.
Okay, that makes sense. Well, that's all lovely to hear. Now, I don't know if you'll be able to answer my last question because it's as I said, a very a growing area of research. But do you know what's next? Do you have any ideas of or plans? What's the next thing you would want to do with FIDA?
So I'm still learning how to use the FIDA efficiently, I think, and the team has been so helpful in helping me bring that online, and especially quickly. But we're only using it really to confirm data that we get in other assays, whether it's a cellular assay or another biophysical or biochemical assay. And so I've recently been talking to the team about doing it in more of a screening method, especially for brand new targets. The AI platform, you kind of want to get a broad sample of all the different chemotypes that you could be trying with a new target. And so it would be cool to be able to screen more compounds in a shorter amount of time using the FIDA. So we've been talking about kind of different assay setups where we're not exactly getting KDs, but we're just kind of getting a yes or no binding. And then we would move forward into like a more of a KD setup experiment to confirm those binders.
Okay, well then good luck with that. I hope it goes well, and thank you so much for sharing your insights.
Yeah, absolutely. Happy to help.